Welcome to this explainer. I am absolutely thrilled to have you here today because we're going to dive into the astonishing thermodynamic machinery that allows plants to move water hundreds of feet into the air. Just imagine a highly integrated network of biophysical and molecular systems operating silently all around us, completely defying gravity without a single moving mechanical part. That's what we're looking at today. We're decoding the advanced mechanisms of plant hydrology. Okay, right off the bat, let's talk about a truly staggering statistic. We naturally assume plants drink water primarily to grow and create energy, right? But the reality is that less than 1% of the water absorbed by plant roots is actually used for biochemical reactions like photosynthesis. Less than 1%. A massive 99% of it is just lost directly to the atmosphere as water vapor in a process called transpiration. Now, that massive evaporative loss isn't a mistake. It actually serves absolutely vital functions, like cooling the leaves and generating the motive force for sap. But this brings us to a massive engineering mystery that we are going to unpack today by following the spatial journey of a single water drop. Really think about it. How exactly does a tree pump hundreds of gallons of water a day from deep underground all the way up to its canopy without a beating heart or a mechanical pump? Section 1. The power of water potential. To understand this journey, the absolute most crucial point is that water movement isn't driven by a mechanical pump. Nope, it's driven by thermodynamic gradients pushing water toward a lower energy state. In plant physiology, we measure this push toward equilibrium quantitatively as water potential. It's actually pretty simple at its core. Water will always spontaneously flow from a region of higher or less negative potential straight to a region of lower, more negative potential. It's entirely driven by the pursuit of equilibrium. What's really fascinating here is how these four distinct forces sum up to dictate exactly where and how fast that water will flow. Think of it like a four-way tug-of-war. You have the solute or osmotic potential, which becomes intrinsically negative as mineral ions dissolve in the water. Then you have the pressure potential, which can be the positive turgor pressure giving a cell its structural integrity, or the massive negative pressure found in the xylem. Next, there's the gravity potential, constantly fighting against the water in tall arboreal species. And finally, the matrix potential. That's the adhesion of water to solid surfaces, like soil particles. All four of these constantly battle it out to determine the water's ultimate direction. Section 2. Roots. The underground checkpoint. So once water actually enters the root epidermis, driven by that lower solute potential created by active mineral accumulation, it has to cross the cortical tissues to reach the central vascular cylinder, which is known as the stele. And it does this using one of three distinct pathways. The apoplastic pathway is kind of like a fast highway right through the porous cell walls, completely avoiding the living protoplast. The symplastic route takes water straight through the continuous cytoplasmic network from cell to cell. And then the transmembrane pathway involves repeatedly crossing the plasma membranes. But here's the catch. Regardless of the pathway the water takes, it eventually hits a physiological wall at the endodermis. Here, we find the Casparian strip. It's this highly suberized and lignified band that acts as a total waterproof barrier. It completely blocks that fast apoplastic highway, forcing all water and dissolved solutes to cross a selectively permeable plasma membrane. It's a brilliant anatomical feature. It acts like a bouncer, allowing the plant to strictly regulate which ions get taken up while preventing accumulated nutrients from just leaking back out into the soil. Okay, so now that the water is finally inside the central cylinder, it faces a monumental vertical climb. And this brings up a fascinating physiological question. How on earth does it overcome that gravitational potential we talked about earlier, especially in trees towering hundreds of feet in the air? Section 3. Upward Ascent. Xylem High Raise. The answer lies in the cohesion tension theory, which is honestly an evolutionary marvel of fluid dynamics. Extreme negative pressure from evaporation up in the leaves pulls an unbroken, cohesive chain of water upward against gravity. Because water molecules have extensive hydrogen bonding, they stick together. That's cohesion. And because they also stick to the hydrophilic, lignified walls of the xylem, that's adhesion, they form a continuous, unbreakable string. As one molecule evaporates at the top, it physically pulls the next one up, transmitting profound tension all the way down to the roots. It's like a giant microscopic chain. But there is a serious risk here. This water is existing under extreme negative pressures in a thermodynamically metastable state. If the tension pulling the water becomes stronger than the tensile strength of the water column itself, it literally snaps. This causes cavitation, It's a rapid nucleation of gas bubbles that creates a localized embolism, effectively blocking the flow of water in that conduit. For the plant, an unmanaged embolism isn't just an inconvenience, it's a literal life or death crisis. This danger reveals a really complex evolutionary trade-off within the plant's plumbing. On one hand, plants have primitive tracheids. These are narrow and compartmentalized, making them incredibly safe and highly resistant to the spread of those dangerous embolisms. But to maximize hydraulic conductance and flow rates according to Poiseuille's law, plants evolved wider vessel elements. Now, these are highly efficient, but their larger diameters and pit membrane structures make them extremely vulnerable to gas embolisms. So extreme efficiency comes at the cost of safety. Astonishingly, though, plants aren't just passive victims to these embolisms. High-resolution X-ray computed tomography has shown us that plants can actively restore their hydraulic conductivity. When we look at this specific refilling rate measured in grapevines, or Vitis vinifera, we see that plants can hydraulically isolate the embolized vessel and actually actively push water in from adjacent living parenchyma cells. This precise influx of water forces the entrapped gas to dissolve, effectively repairing the broken water column. It is a fully active, living repair mechanism. Section 4. Stomata and the sky. Following our water column all the way up to the canopy, we reach the exit point. This is the mechanical cascade in action, showing exactly how turgor pressure acts as a hydraulic lever to open the stomata when conditions like light and internal CO2 are favorable. First, proton pumps hyperpolarize the guard cell membrane. This voltage shift opens channels that allow a massive influx of potassium. The buildup of potassium, chloride, and malate drops the solute potential, which vigorously draws water in via osmosis. This inflates the guard cells, supercharging their turgor pressure and causing their radially mycelated cell walls to bow outward and open the pore. Conversely, when water is scarce, the plant deploys the hormone ABA, or abscisic acid. You can think of ABA as an emergency handbrake. It triggers the rapid exit of those potassium ions and anions, leading to an immediate loss of turgor pressure. The guard cells completely deflate, the stomata close shut, and the transpiration rate is drastically reduced to save the plant from drying out. Section 5. Aquaporins, the micro gatekeepers. Finally, let's zoom all the way down to the subcellular level to meet the microscopic machinery regulating this entire global flow. These aquaporins are the true gatekeepers. They are integral membrane proteins, like pips and tips, that provide plants with the ability to rapidly and reversibly modify their membrane hydraulic conductivity in response to environmental cues. They aren't just static open pipes. They are highly regulated valves, capable of passing water and even small neutral molecules like CO2 and silicon, exactly when and where the plant dictates. And what's really wild is how the opening of these microscopic gates is controlled. Intricate intracellular signaling cascades use calcium-dependent protein kinases to recognize specific motifs and phosphorylate the aquaporins, keeping them in an open conformation. And get this operates on a distinct circadian rhythm. The phosphorylation state naturally peaks right around midday, perfectly timing the opening of the floodgates exactly when midday transpirational demand from the sun is at its absolute highest. But these gatekeepers are also incredibly sensitive to danger. If the roots get flooded and waterlogged, oxygen levels absolutely plummet. The plant's cellular respiration shifts to anaerobic fermentation, generating organic acids that cause cytosolic acidosis, which is a rapid drop in cellular pH. This sudden low pH acts as a rapid response physiological break. It triggers the structural closure of the aquaporin pores within minutes, slamming the doors shut on water uptake long before cellular ATP depletion becomes a fatal issue. We started today by just looking at a simple drop of water. But when you realize that plants are operating as these highly advanced, actively regulated hydraulic engines, balancing thermodynamics, evolving complex pressure chambers, and managing microscopic circadian gatekeepers, you really have to ask yourself, what other thermodynamic miracles are happening invisibly beneath our feet, just waiting to be understood? Thanks so much for joining me on this explainer. Keep wondering and keep learning.